All right, well, everybody can come on in, and I appreciate you all hanging around. Settle down. Oh, there's a lot of people still out there. Oh, Dr. Powell. Okay, we have everybody we need.
Oh.
I'm Susan Poteet. I'm one of the KidneyCAN team, and I wanted this chance to get up and introduce this session. It's the first time we've done a biotech session. Like all the other members of the KidneyCAN team, I have a cancer story. My father-in-law was diagnosed with kidney cancer in 2001, and my husband in 2007. Back then, kidney cancer was a desert island. Nobody was researching it, nobody was talking about it, and there really weren't any treatments. So this day, when we really reached one of the dreams of KCRS of getting small industry who chose kidney cancer for research instead of another cancer. We patients don't always remember that there are lots of cancers. So we're glad to have you here, and I'll turn it over to Dr. Hummers and Dr. Powell.
Great. Thank you so much, Susan. Susan is an invaluable member of KidneyCAN, no doubt about it. So this is, I think, a very special session, really with a focus on, our smaller biotech partners and champions, if you will. They play an incredible, importantly role. They often champion new molecules, new therapies, etc. So I think great. Thank you so much for coming and giving us your time to talk about things. So without further ado, I would like to invite Dr. Dimitry Nuyten from Arcus Biosciences to talk about the exciting molecule. Thank you so much.
Thanks, everyone, and thanks for the opportunity to present. My name is Dimitry Nuyten. I'm a radiation oncologist by training, and about 16 years ago I decided to, let's say, move to industry to do drug development instead of clinical radiation oncology and clinically focused research on the academic side. And since 2 years I'm the Chief Medical Officer for Arcus, a small biotech company based in the San Francisco Bay Area. The title of my talk is pretty straightforward. Casdatifan, formerly known as AB521, a HIF2α inhibitor in development for clear cell renal cell carcinoma. Casdatifan is an orally bioavailable small molecule inhibitor of HIF2α. It has a high potency, as demonstrated in preclinical models, and I won't go through all the details, but all the details are listed on the right side of the slide. And not so much talked about, but in early drug development, clinical pharmacology is a very important part of a phase 1 trial, and especially for HIF2α, as our head of research, who is a medicinal chemist, actually says binding to HIF2α was not easy to create a good small molecule inhibitor, but it took 2 more years of optimization of PK. There's a lot of challenges with a targeting transcription factor and then making a molecule that behaves well in the clinic so we can optimally develop the molecule. Our first inpatient study is the ARC-20 study. We have tested this molecule in normal healthy volunteers, generating valuable pharmacokinetic and pharmacodynamic data. And the first inpatient study is called ARC-20. It's ongoing. On the left side, you can see the dose escalation steps. We started at 20 milligrams. We went up from 50 to, say, 50 BID. And 50 BID is only done for capsule limitation reasons. We started with 10 milligram capsules. We did not think it was reasonable to give patients 10 capsules early in the day. We have since then transitioned to tablets. The tablets are 25 milligrams and bioequivalent. So I refer to this cohort as 100 milligram, and moving forward, everything is 100 milligram in 25 milligram tablets. And then we have escalated further to 150 and 200. We are currently exploring 200 up till and including 150 milligrams. We have not seen any dose-limiting toxicities. We have started expansion cohorts. The dose escalation did allow patients with other histologies than kidney cancer, and it also included non-clear cell kidney cancer patients. The expansion cohorts are all in clear cell renal cell cancer. We have different dose levels, as you can see, 50, 100, and 150. 100 milligram was done first, then followed by 50, then by 150. They're not randomized, they're done sequentially. And the short answer, why so many dose levels, this is really in talks with the FDA for Project Optimus for dose optimization, generating a wider range of efficacy and safety across different dose levels. And our latest cohort is the combination with cabozantinib that just started enrolling. A little bit about why we think Casdatifan could be differentiated, and obviously the statements I'm making are research hypotheses. They have to be shown in the clinic. We start with the normal tissue or normal physiology biomarker or pharmacodynamic marker EPO that is now well known because of belzutefan's work. Belzutefan as a reference, based on the label, has about a 60 to 65% reduction of EPO. We think that not all EPO is regulated by HIF2. You can probably get to about 80 to 90% inhibition. We've shown from dose levels from 20 to 150 that we are now at about 85% EPO reduction. And you can see that modeled out in the curve on the right side. And what I always try to explain when people ask about is I think it's important to have a pharmacodynamic biomarker, but I do think that tumor physiology or pathophysiology is different from normal tissue, and therefore we think, quote unquote, pushing the dose, if safety allows it, makes sense beyond EPO inhibition, because it's hard for me to imagine that, let's say, what you can achieve in normal kidney tissue with vascularized organ versus a tumor environment, that having more drug available in the circulation could theoretically be beneficial and therefore have potentially higher tumor efficacy. And therefore we're interested in, let's say, going up in dose. Even EPO seems to be maxed out. This shows more about the PK profile and why we think this is important. As I said, it's hard to create a well-behaved pharmacokinetic molecule for HIF2 alpha inhibition. You want a half-life that is not too short because you don't want to do more than daily dosing for patients. You want a half-life that is not too long, because if you have a very long half-life, first of all, you cannot do dose interruptions to manage toxicities like anemia and hypoxia. A half-life of multiple days would mean that patients, if they stop the drug, they would still have days and days of active drug on board. Our half-life is about 20 hours, which we think is fairly optimal for daily dosing, but also means that after a few days, the levels go down fast in the circulation if you want to interrupt the drug. We also have linear PK. That also is the reason why we think the dose testing multiple dose levels makes sense, and it's relatively straightforward. At a dose that is 5 times higher than the 20 milligram starting dose in our trial, the exposure level is indeed 5 times higher. It's a completely linear PK, and the first data from 150 shows the same. We still have linear PK going up to 150, meaning if you go from 100 to 150, you will see 1.5 times higher relative drug levels. I don't have time to go through details, and we don't have that much detailed data in the public domain, but the safety data is as expected with anemia, fatigue, and hypoxia seen. This is the data from the dose escalation, and I'll move fast because I'm running out of time. The little bit of data that we've talked about so far is the dose escalation. We've seen multiple patients with prolonged stable disease, and I can mention that after the database cutoff, the patient that was on study for 14 months with clear cell renal cell cancer had a response starting at right after 14 months. We have seen responses also coming in faster than much faster, including patients sometimes having a response at the first scan already. The dose expansion cohort of 30 patients, we are planning to present the detailed data. This is a relatively old data cut. We are planning to present the detailed data at a medical conference later this year. And then the last slide, I'll go fast. The ARC20 study, I mentioned it. The Stellar-09 study with Exelixis has been mentioned earlier today, where we are combining with Sensilimab, and we are preparing our phase 3 trial for a start in the first half of next year that will be named PEAK-1 in the post-IO setting, meaning post-Pembrolizumab in the adjuvant setting or post-checkpoint inhibition in the first-line metastatic setting. Thank you.
So we will have first 5 presentations, and then there will be a question answer session. So next speaker is?
Next we'll go to Lou Gan, who's from Scholar Rock, discussing SRK-181.
I think that one moves forward. Where? This is the player here.
So thanks for the invitation to speak. So today I'm going to talk about our drug, SRK-181. It's well established that the TGF-beta pathway is involved in the tumor immune escape and the resistance to the checkpoint inhibitor treatment. SRK-181 is a selective latent TGF-beta1 inhibitor. So it basically binds to the whole complex of the latent form, and it prevents the release of the activation form. So our drug is designed to increase the therapeutic index by two specific ways. First, the selectivity specific to the beta-1 isoform is designed to avoid the off-target toxicity has been seen in beta-2 and beta-3 isoforms. Second, the context-independent inhibition, of the so the context-independent inhibition allows SRK-181 to target all the components of the tumor microenvironment, including the tumor-associated macrophage, the MDSC, and the regulatory T cell, etc. So therefore, it could maximize the efficacy potential. So the phase 1 study is a typical phase 1 study. It started with a dose escalation with monotherapy phase and a combination treatment with Pembrolizumab, followed by the dose expansion. So in the dose expansion, this combination treatment, SRK-181 with Pembro, was tested in 6 specific tumor types. One thing I want to highlight is we're targeting, basically patients are required to receive anti-PD-1, and they are anti-PD-1 resistant. Here's the data. Since this is the kidney cancer conference, we'll focus on the ccRCC cohort. 30 patients were enrolled, and the median prior line of the therapy was 2. And as I mentioned, all the patients received anti-PD-1. And you can see around 30% of the patients received more than 2 lines of the anti-PD-1 therapy. And all the patients are anti-PD-1 refractory, representing as a best response to the prior anti-PD-1 was stable disease or disease progression. And all the patients' disease progressed from the most recent anti-PD-1 treatment. I also want to add 20 out of the 30 patients also received a TKI treatment. And if you look at the IMDC score, majority of the patients were intermediate and poor. All the 30 patients, among the 30 patients, 4 are still ongoing. And for those who discontinued, around 16% of them were discontinued due to treatment-related AE and mainly the skin-related toxicity. SRK-181 was well tolerated as a monotherapy as well as a combination treatment. Here I'm showing you all the Part B patient safety data from the 78 patients. The most common treatment-related AE were rash, pruritus, and you can see only have 1 treatment-related grade 4 AE and without any treatment-related grade 5 AEs. The treatment-related SAE, over 2% was pemphigoid only. As I mentioned, though this patient is heavily pretreated and has anti-PD-1 refractory, and the response rate is around 23%, including 1 PR patient and 1 CR patient and 6 PR patients. So if you look at the butterfly plot on the left, the light blue bars on the left representing the prior anti-PD-1 treatment. And on the right side, the green colors were our combination treatment. So the combination treatment not only changed the non-responders to the responders. If you look at the other patients, they stayed on the combination treatment longer than the prior anti-PD-1 treatment. Here's some biomarker data. The biomarker data really proved the proof of mechanism of this drug, particularly in human. So paired biopsy were collected from baseline and post-treatment. On the most far left panel here, you can see the combination treatment increase the CD8 positive T cells across multiple tumor types. And in the middle panel here, we measure the CD8 positive T cell along with the granzyme B. So you can see the CD8 positive T cell were activated in patient who has PR or stable disease, which were orange and green, gray color, but not the PD patient. At the same time, if you look at the T cell activation, it seems correlated with the shrinkage of the tumor size pretty well. So taking all the data together, basically it shows the combination treatment creates a pro-inflammatory microenvironment. I'll do quickly. So this is the ccRCC cohort. We're looking at the baseline Treg level on the left, and the left is the CD8 positive T cell, and the right is the Treg. So you can see the patient with the high baseline, either CD8 T cell or Treg level, you see a better clinical response. And if you look at the response rate, it's also increased. So to conclude, the anti-tumor activity in this combination treatment, particularly in anti-PD-1 refractory patient population, are promising. And the drugs are tolerated pretty well, and the biomarker results also show the proof of mechanism and some potential patient selection strategy. It warrants the further investigation of this compound. Oh, lastly, I just want to say thank you for the patient, for the family, and PI and study centers.
Thank you so much. So next speaker is Alex Azrilevich from Synthekine.
Thank you very much to the KCRS leadership for selecting STK12 to be discussed in the session and for the invitation. Alex Azrilevich, I'm in the clinical group at Synthekine. I came to Synthekine from BMS, where I was in development there for about 13, 14 years, and was the bladder lead for some time. So GU oncology is very much near and dear to my heart. And so we're very much glad to be making some inroads potentially with engineering IL-2 to potentially provide some benefit. And so today I'm discussing STK12, which is a first-in-class alpha-beta IL-2 receptor biased partial agonist in advanced solid tumors. So to borrow from David Braun today, if you want to engineer the right kind of cytokine into a therapeutic, you want to make sure to maintain the gas pedal, which is what nature, basically created cytokines to do. But you want to try to add as much of a steering wheel as you can to target the right kind of lymphocyte population. And cytokines, as we know, are by nature very pleiotropic. They activate and expand multiple different kinds of lymphocytes. And from the standpoint of Synthekine, the right lymphocyte is really the antigen-activated CD8 T cell. That's where more than likely your tumor-specific T cells are going to be. And that's what STK12 is designed to do. It's designed to particularly bind to lymphocytes that express a high level of the trimeric IL-2 receptor that has all three subunits, the alpha, the beta, and the gamma. That is, and the reason that makes sense for antigen-activated T cells is that when a T cell sees antigen, it significantly upregulates the trimeric IL-2 receptor, almost 3 log. And so to take advantage of that particular event, we've designed STK12 with reduced affinity at the gamma subunit to selectively bind to trimeric expressing lymphocytes. And so that is sort of the opposite of what the non-alpha IL-2s have tried to do. I think there was an effort to avoid the alpha subunit to try to spare the vascular leak type of toxicity. But so far, there's been a number of programs, and some of them are still in the clinic, so I think proof of concept is still outstanding, but there's been some disappointments in the field. And of course, that is all different from what aldesleukin did, which was a recombinant human IL-2 non-selective cytokine, which showed great promise in a small percentage of population, but had highly intolerable toxicities. So we're first in class with this approach, and we're really looking forward to kind of driving forward the idea of alpha targeting as a way to induce anti-tumor effect. And this is similarly to what I just kind of described, just a cartoon depicting that the toxicity that you see on the bottom, the vascular leak syndrome in our hands in mouse studies as well as in cyno was really driven by NK cell activation and expansion. And those, of course, express the dimeric, largely form of the IL-2 receptor. So we did not want to target those populations, and so the efficacy is going to be driven by antigen-activated CD8 T cells, which significantly, of course, upregulate the trimeric IL-2 receptor, which STK12 targets. And I'll be describing the preliminary data from the phase 1A monotherapy dose escalation. STK12 is a pegylated, subcutaneously delivered molecule. We've gone through monotherapy and combination escalation with Pembrolizumab in advanced solid tumors. We're currently in single agent expansions in RCC and non-small cell lung. Today I'll be covering data from monotherapy dose escalation in 47 patients. The majority of the population was pre-treated with a prior checkpoint inhibitor, and the common tumor types were non-small cell lung and clear cell RCC. With respect to safety, the table there describes the most common treatment-related toxicities, and what we saw was most commonly rash, fatigue, injection site reactions, GI toxicities, and arthralgia. What we did not see much of at all are your kind of traditional capillary leak kind of treatment-related toxicities. So very few patients had hypotension, pyrexia, peripheral edema. No patients had CLS. No patients had adverse events of LFT increase. And for us, that was a major first domino to fall of targeting alpha, in fact, does not induce your vascular leak. So we're sort of on the right track. And most of our toxicity was reversible and manageable with standard approaches of dose hold, dose reduction, supportive meds, including in some cases oral steroids. There were no DLTs, and the majority of tox was either grade 1 or 2. The dose that we ended up choosing for monotherapy dose expansion was 2.25 milligrams flat dose Q3 week, and that's where we are right now in both lung and RCC expansions. Some preliminary PK/PD data. So this is from our Q3 week monotherapy patients. It's a little bit difficult to see all the curves here, but this is across 5 dose levels. We have a dose proportional increase in the area under the curve for our PK exposure. The half-life of the molecule is 4 days, which was once again pretty exciting for us. Of course, aldesleukin is an hour, and the majority of non-alphas are somewhere around 12 hours or less. And so this supported the ability to deliver this molecule in a Q3 week fashion. And some of the proximal pharmacodynamic markers, we looked at STAT5 signaling and which cells are actually driving the STAT5 signaling. And in fact, what we saw is that the CD25 positive cells, the sort of dark blue and the aquamarine, that's your CD25 positive CD8s and CD25 positive CD4s are driving the majority of the STAT5 signaling. So this molecule is pretty specific for CD25 positive or cells that express the trimeric IL-2 receptor. And our peripheral interferon gamma looked to be dose dependent with nice step up across dose levels. That light blue is 2.25, and that's the dose that we ended up choosing to go into monotherapy dose expansion. And I'll go pretty quickly because I'm running out of time here. We also looked at broad sets of lymphocytes, and the idea is that because this is supposed to be targeted to antigen-activated T cells, we should not be expanding broad sets of CD8s or TCONs, and that's in fact what we see. We don't see really an effect in broad CD8s and TCONs, modest effect on Tregs, and no expansion on NK cells. But if you dig into the specific subpopulation of CD8 T cells that are antigen activated, we have a nice dose-dependent increase. Preliminary monotherapy efficacy data does show single-agent activity. This is across different tumor types, across different doses. We have 3 responders in this data set, renal, clear cell RCC, head and neck, and non-small cell lung. And in the in our subset, that is actually 1 to 2 prior lines. Dr. Pell, this is the subset that you referenced earlier with respect to phase 1 studies. In this subset, we have a pretty decent single agent activity here and earlier signs of durability. And this is the last data slide that I'll leave you with. This is an additional responder that we saw from our phase 1B data set. We're still accruing in this data set. This is a patient that came on after having progressed on Nivo-Ipi and Cabo-Nivo. This is an RCC patient, came on with innumerable lung mets and a large primary renal mass. It's sort of difficult to see, but you see there what happened after 12 weeks of STK12 Q3 week. There was an 85% reduction in target lesions. Most of those target lesions disappeared and a number reduced. Patient had a very large primary renal mass, and so which was non-target. If you count that, the overall response was 42%, but the official target response here is 85%. And this patient progressed at week 24. So we're encouraged by the initial signs of PK/PD safety and efficacy that support that the profile here supports the strategy of targeting the trimeric IL-2 receptor that includes the alpha subunit. And so far the profile looks different than what you would see with aldesleukin or non-alpha IL-2s, and we're encouraged in moving forward into expansions in lung and RCC.
That was great. It's a pleasure to introduce dear friend and colleague Chet Bohac from Xencor. Chet, thanks for joining us.
Thank you. And thank you to the organizers for this opportunity to present today. I'll be presenting on the 81901 phase 1 study, evaluating a novel bispecific antibody targeting ENPP3 in clear cell renal cell carcinoma. ENPP3 is a naturally occurring enzyme. It converts ATP to AMP. It's found in basophils. And its action is to prevent allergic reactions from evolving into anaphylaxis. ENPP3 was identified by TCGA and confirmed by immunohistochemistry as a tumor target. ENPP3 is highly expressed in clear cell renal cell carcinoma, and you can see that data in the graphics on the right. Because of that, clear cell renal cell carcinoma was selected as the tumor type for this phase 1 study. Other tumor types that express ENPP3 include papillary renal cell, lung, and colorectal cancer. On this slide shows the bispecific 2+1 format of the 819 study drug. There are 2 arms in blue that bind to ENPP3 and 1 arm that binds to CD3. All 3 of these arms need to be bound and engaged to induce cytotoxic activity. 819 is selective for cells that highly express ENPP3 over normal cells that have low expression of ENPP3. This slide shows the difference between the 2+1 819 format versus other bispecific antibodies that have a 1+1 format. In the 1+1 format, there's broad reactivity. There's only 1 arm that binds to ENPP3, whereas the other arm binds to CD3. So it can bind to low expressing normal cells, as well as high expressing tumor cells. The 2+1 format is not able to bind to low expressing cells very well. It just kind of drops off. It needs both arms to be bound. So it needs a high expressing cell so that those ENPP3s are close together, can bind a T cell and be activated for cytotoxicity. This is why there's an advantage for this 2+1 format for anti-tumor selectivity. This slide shows a tumor model in immunodeficient mice. These mice were engrafted with ccRCC, which is known to highly express ENPP3. It was allowed to grow for a couple weeks, and then engrafted with human PBMCs. They were then treated with PBS and 819, and then the graphics on the right show that the tumor volume was suppressed. This information led to the development of the phase 1 study. The objectives of this study are: primary objective is safety and tolerability, secondary objective PK and preliminary tumor activity. A priming regimen is used with this bispecific to mitigate the incidence of CRS. A priming regimen has a very low priming dose, followed the following week by a step-up dose, and then the third week, an efficacious target dose. This regimen is dosed weekly. The inclusion criteria are for relapsed refractory patients with at least one prior checkpoint and one prior VEGF TKI. There's no limit on the number of prior therapies. Patients with clinically unstable disease or poor organ function are excluded. And down in the lower left shows that we're doing both IV administration cohorts as well as sub-Q administration cohorts. And once we have a biologically active, safe, recommended phase 2 dose, we'll open a dose expansion, which we hope will be opening by early next year. And monotherapy will be explored in ccRCC, and then we'll also be doing combinations with checkpoint inhibitors, TKIs, potentially belzutifan, and other novel therapies, and other solid tumor types that express ENPP3, such as papillary renal cell carcinoma. Thank you for your attention.
Thank you so much. The last speaker for this particular five-count presentation is Dr. Levi Gray-Rupp from ArsenalBio, and I hope we're going to get our technical issue resolved that we have here on the screen.
It's my fault. The slides were massive. I didn't do a good job shrinking them, so apologies.
Okay, I think it's getting there.
While they're popping up, maybe I'll give a brief introduction to ArsenalBio. So again, thank you to the organizers for the invitation and the opportunity to speak. ArsenalBio is a company based in South San Francisco, and it was founded with the goal of developing next-generation CAR T-cell products to cure solid tumors. Today, I'll be describing the development of AB2100, which is our second clinical stage program, and it's designed for the treatment of clear cell renal cell carcinoma. So AB2100 is a somewhat complicated product, so bear with me here as I step through the components. It contains 4 features or modules, as we refer to them, all of which are designed to increase the selectivity and the potency of the cells in RCC. So the first component, is there a pointer by chance? We'll see. Thank you. So the first module is a logic gate. This is a 2-receptor system that I'll explain in greater detail on the following slides. But in brief, the first receptor recognizes PSMA on the tumor vasculature and then induces expression of the CAR molecule here shown in red, which recognizes CA9 on the tumor cells and mediates cytolysis. The next 2 modules are shRNAs that target TGF-beta receptor 2 and FAS respectively within the T cells. Knockdown of these proteins within the therapeutic cells reduces their susceptibility to immunosuppression in the tumor microenvironment. The final module is a synthetic pathway activator, or SPA, which signals through the JAK-STAT pathway to increase T cell persistence, resistance to exhaustion, and anti-tumor potency. I mentioned that the CAR target is CA9. We think CA9 is a really compelling target antigen in RCC because it's highly expressed on the vast majority of tumor cells in the vast majority of patients. You heard yesterday that it's the definitive histologic marker for clear cell. The challenge with CA9 is that previous efforts to target it have been limited by on-target off-tumor toxicity due to expression in normal tissues, primarily the bile ducts of the liver. And this is where the logic gate technology that we've developed comes in. And so the hypothesis here is that we can restrict the activity of these cells to the tumor environment by requiring the presence of 2 antigens in proximity. And the second antigen here is PSMA. So we know that RCC tumors are highly vascularized, and it turns out that those vascular endothelial cells frequently express PSMA at a fairly reasonable level. So how does this work in practice? In the basal state, there's no expression of the CAR. The CAR is not expressed on the cell surface. So if the therapeutic cells encounter healthy tissue that expresses CA9, nothing happens because there's no CAR around. However, when the cells traffic to the tumor, they encounter PSMA on the tumor vasculature. This first receptor undergoes proteolytic cleavage and turns on transcriptional expression of the CA9 CAR. So the CA9 CAR is expressed, comes to the surface, and can now mediate cytotoxicity against the CA9 positive tumor cells. And then in the absence of the PSMA signal, this CAR will decay and be degraded, and the cells will return to the baseline state. We've demonstrated the selectivity of the logic gate in multiple preclinical assays. Here I'm describing what we call the dual flank tumor model. We implant mice with 2 tumors on the contralateral flanks. The first tumor expresses both PSMA and CA9 and is supposed to represent the RCC tumor. The second tumor expresses CA9 alone and represents the healthy tissue. You can see that upon treatment with AB2100 in the red curves here, you have robust anti-tumor activity against the flank that expresses both PSMA and CA9. In contrast, in the tumor that expresses only CA9, there's no detectable anti-tumor activity beyond the control cells. So this suggests that the T cells are highly selective for tissues where both PSMA and CA9 are expressed in close proximity. We've similarly validated the function of the other components preclinically. Here we're showing that AB2100 cells in red are resistant to fast-mediated apoptosis, whereas cells that lack the shRNA, the fast shRNA, are susceptible. In a similar fashion, addition of the TGF-beta receptor shRNA increases the anti-tumor efficacy both in vitro and in vivo in models where TGF-beta mediates T-cell-based suppression. Last but not least, the synthetic pathway activator, or SPA, provides a significant increase in anti-tumor potency. So here in the blue curves, we're showing treatment with AB2100. One thing I would note is that these are quite large xenografts. These are about 300 to 400 cubic millimeters, and the dose is very low here. We're treating with 50,000 CAR T cells. We get complete and rapid regression. And interestingly, if we then remove the SPA component, shown here in the green curves, we have a significant decrease in the anti-tumor efficacy. So we think this is a key driver of the potency and activity here. As would be expected, if we benchmark this against a conventional CA9 CAR, we see that AB2100 significantly outperforms a standard CAR T cell. I don't have time to go into great detail today, unfortunately, around manufacturing, but I will note 2 things. This is an autologous process, and importantly, it's a non-viral CRISPR-based targeted knock-in. So we don't use a viral vector. We use Cas9 to make a DNA double-strand break and insert our transgene cassette. This is crucial because the amount of DNA that's required to encode all of these functions is larger than can be provided by a viral vector. So this is really a foundational technology that enables us to do this complex engineering of T cells. And finally, I'm very pleased to report that we are actively enrolling a phase 1 dose escalation study in patients with ccRCC who progressed after checkpoint inhibition and a VEGF-targeted therapy. The phase 1 is a standard 3x3 dose escalation with backfill cohorts, obviously looking to assess safety and tolerability and identify a recommended phase 2 dose. As I said, it's an autologous therapy, so we leukapheresis patients. It takes about 28 days to manufacture and release the product, return it to the centers. Patients undergo standard lymphodepleting chemotherapy and then receive their treatment. I think the last thing I would mention is that many folks in this room have been very integral in the design and execution of this early stage of this study. So we're very grateful to all of you, as well as the patients who have chosen to participate, and look forward to providing further updates in the future. Thank you.
Great. So now we are open for questions.
Do you go?
Great. So, Dimitry, you went through this slide pretty quickly, but can you describe the phase 3 trial again?
Sure. So I don't think I'm officially allowed to comment on whether or not it's Cabo or Sansa, but I can comment more. So it's what we call the post-IO setting. So patients who progress after adjuvant therapy with Pembrolizumab, we are debating the exact interval that's under discussion. And then patients who have gotten Ipi/Nivo or a checkpoint inhibitor, a PD-1 plus VEGF-TKI, they are all eligible, with the exception of patients who got the combination TKI of choice in the trial before, and then it will be placebo-controlled, meaning, of course, everybody gets active standard of care, but we have a placebo for Casdatifan which we think is very helpful for the trial integrity and the optimal safety profile.
So it's essentially TKI plus your drug
Correct.
versus TKI.
Yes, our first development option is a combination strategy. That's correct. Yes.
Okay. And then Alex, I was just curious. It was interesting presentation. It was interesting to see no lymphopenia and no lymphocytosis, which suggests that it's no peripheral activation of NK cells or other cells. But do you have any intratumoral data showing activation of the cells that you wanted to activate?
Yeah, it's obviously something that we're interested in developing. We don't have data yet on the intratumoral setting to look at the pharmacodynamics at the tumor level. We're actively working on that.
Over here, Alex. So the strategy, so Tregs have attack CD25 constitutively on their surface. So how does that work into your therapeutic strategy? I mean, are you not going to activate Tregs with your?
Yeah. So so preclinically, we initially, obviously it's a central question for us. So preclinically, we saw that in the tumor, despite activating modestly Tregs, we had a vastly increased ratio of antigen-activated CD8 T cells or effector cells versus Tregs. And so that allowed us to sort of have some confidence to move it forward to the clinic. We don't have tumor data yet, but in the periphery, if you look at the level of expansion of antigen-activated CD8 T cells, there are CD38, Ki67 CD8s. They sort of double or triple the expansion that we have of Tregs in the periphery. For the non-alphas in particular, which are designed to avoid alpha and avoid Tregs, you do see sort of a 2 to 3-fold increase in Tregs as potentially a compensatory or potentially an off-target effect as well. So I think it's hard to completely, get rid of any Treg activation. But so far, we're seeing that clinically we're able to see responses even in the setting of some modest Treg activation, and largely we think that's due to a much higher activation of antigen-activated T cells.
Thank you.
Over there. This is a question for Dr. Gray-Rupp. You showed the mouse data showing that it's really only the PSMA expressing. So I'm wondering about cycling of the T cells out of the tumor. So I guess 2 questions. One, did you actually look at the contralateral tumor to see if some stuff was actually getting in, even if it wasn't actually having an effect on the tumor size? And the second question is do you consistently need PSMA there to actually have transcription of the CAR, or as soon as you leave, does the PSMA gone and the CAR goes down? And what's the time course?
Yeah. Hello? Okay. Excellent questions, and somewhat complex. A couple short answers is that we have looked in the contralateral tumor, and we don't see any sort of meaningful CAR expression to the best of our detection. But again, certainly, the tumor is growing, so the sensitivity in that assay may be a little lower. As far as how much PSMA do you need and for how long, right? That has been very challenging to really quantitate. As you can imagine, the more PSMA you have around, the more CAR expression you get, and then the more sustained expression. The other variable is that CARs, when they recognize antigen, will be internalized and degraded. And so if you have high levels of target antigen CA9 on the tumor cells, that's going to also affect your kinetic, essentially. And so I think what we've shown, I didn't show you the data here preclinically, but we've shown that you can prime off of one cell and transfer those cells and have retention of anti-tumor activity. You can kind of do mixed co-cultures, et cetera. It's a little challenging to study in vitro, with shear stress and flow, et cetera. But we think that there's a reasonable duration there without it being excessively long to risk toxicity from recirculation.
Alex, so great talk. But I want to know, what are you doing? And this is what I mean, is that in order for your cytokine to work, you have to have activated T cells, right? So there has to be activated T cells. And so I think it's fascinating that in these PD-1 refractory patients, you have activated T cells. So if there are tumor-specific activated T cells, what are you doing to them? And why is it that you give your cytokine and it works, and you give anti-PD-1 to activated T cells and it doesn't work?
Yeah, so it's a great question. You know, I can tell you we've done we submitted an abstract to SITC on our translational data to look at clonal expansion. And, you know, we're seeing that clones that do not exist at cycle 1, day 1, by cycle 4, day 1, are there, and they expand. And you know, with work, you know, through work with Adaptive, you know, a number of those clones are part of their sort of library of TCR that are specific to cancer antigens. So, you know, I don't have a great, you know, answer to your question, except that we are observing activation and expansion of presumably tumor-specific clones that may even not register at cycle 1, day 1 with single agent STK12. That's as much as I can say about the data.
Yeah, so actually, just one last comment is that, so that's actually very encouraging. I'll tell you why. Because as someone interested in immunotherapy, one of the things I fear the most is, what if the cells aren't there? You know, you can't do anything. But you might say that your data is suggesting that, you know, even for other companies and ideas, that that even when you don't see the cells, they're there. And so that's an important point.
And my particular bias was that, you know, we really need to see, at the outset of the programs, we need to see some precursors. But we have found that in patients who don't have these precursors at baseline, I mean, we're still able to kind of elicit and expand some clones that we think are tumor specific that weren't there at cycle 1, day 1.
Great, thank you.
So question to Levi about, interesting that you are targeting with your CAR, with your autologous CAR, CA9 and PSMA, knowing that, you know, PSMA is not as highly expressed on clear cells as CD70. What was your rationale for going targeting PSMA and C9 instead of CD70 and CA9?
Yeah, absolutely. So the first piece is the selection of CA9 over CD70. So in our experience, CD70 is quite a bit more heterogeneous, right? And that's reflected in, you know, for instance, Allogene has moved towards a selection strategy in their study. So we think CA9 is more homogeneously expressed in a larger fraction of patients. And then the second piece is that, yes, so the PSMA, it's actually not expressed on the tumor cells at all. It's purely on the tumor vasculature. And so it essentially, you know, the nature of our technology is something that allows us to then target those two in combination if they're in proximity, in general proximity, essentially. And so, you know, the identification of PSMA came through sort of a extensive target identification to see what could pair with CA9 and restrict the activity away from sites of normal CA9 expression.
So my question's also for you. So I just wondered, recently there's been some PSMA expression in the brain with some neurotoxicity, and I just wondered how you're going to mitigate that, and is there any kind of a switch receptor or mitigate it?
Yeah, excellent question. We do not have any sort of suicide switch or any depletion strategies there. I think that data is complex, I would say, some of the PSMA expression data there. I think one thing is that, you know, basally, we don't know how frequently the CARs really get to that tissue. But if you also think a little bit about the dual flank tumor model, you know, what that suggests is that you can prime in one location, but by the time you transit to another location, it's unlikely you have enough CAR remaining to cause damage. And so we would hypothesize that that would be similar in that instance as well.
I think we believe that it's in the neovasculature, not actually in the tumor.
It is exclusively in the neovasculature.
Yeah.
Yes. Sorry if that hasn't been made clear.
Okay.
I was trying to emphasize that. Yes, I apologize. Okay. Yep. So our IHC is very consistent. Our RNA-seq is entirely in the vasculature.
And so then you're just trafficking the cells.
Yep. On the way in, the cells
Got
encounter
it.
PSMA and then express.
Okay.
I will say, We do have some data that suggests that it's expressed in pericytes, which would then line and sort of be on the adjacent side of the tumor cells themselves. So that may contribute, but it is very clearly it's not on the neoplastic cells, it's on the vasculature.
I mean, your CAR are double, so it's all like a bispecific, PSMA and CA9. What if some of them gonna lose some of these antigens just because they're not perfect? What if some that lose PSMA and CA9 only and go to the bile duct?
So are you referring to antigen escape or?
Right, antigen escape,
yes,
losing.
yes.
I mean, nothing is perfect.
Right,
You're
absolutely.
infusing many CARs.
So, and I apologize, I went very fast today on the mechanism. So we don't require both antigens to be expressed on the tumor cells themselves. It's not a true AND gate, where other groups have described true AND gates. So you have to first recognize the priming receptor PSMA, and then there's a cleavage event that leads to transcriptional induction of the CAR. And so then the cells can still traffic and move, and they'll have upregulated the CAR expression at that point. So they just have to encounter PSMA and CA9 in some temporal window, essentially. So there shouldn't actually be any selective pressure on PSMA expression within the tumor cells, certainly on CA9, and that's something that we think about, but you don't need both antigens to be expressed at high level on the same cell.
Yeah, but my question was, with CA9 being also in some normal part like the liver, biliary tract, and is the fact that it's against PSMA only save you from this problem, the potential liver issues?
That's the working hypothesis, right? Is that we'll restrict that activity preferentially to the tumor, right? Now again, will it be 100% protective? We have to run the studies to find out. But again, based upon the preclinical data, we would hypothesize that it would have a significantly increased therapeutic index, certainly relative to a constitutive CAR.
Mm-hmm.
So it sounds like, on that a little bit. So it sounds like you actually have a little bit of a bystander effect. You must have one, because as they transit through the endothelial cells, they get activated by the PSMA on the endothelial cells, but they maintain the CAR expression long enough to actually do damage beyond the endothelial cell. Have you mixed CA9 positive and CA9 negative cells in the same tumor? Obviously, on the other side of the animal, you don't see the anti-tumor effect. But it would be interesting from a perspective of losing potentially some antigen, having some heterogeneity in the same tumor, you still have kind of like a mini field effect, if you will.
Yeah, we haven't looked at this carefully. I would assume this would be similar to any sort of CAR T cell that's going to make a lot of cytokine locally and potentially do some bystander killing, but the cells haven't been specifically designed for that, and we haven't strictly benchmarked that against other strategies.
Taking the other part of your compound, the SPA, I thought that was pretty provocative data. It seemed like your CAR T did not have a lot of efficacy without the SPA. The SPA is a critical component. I wonder whether adding SPA to other CAR T programs that have been developed, obviously that wouldn't be in the interest of your company unless you have the licensing, but that, in the CD70 CAR T, would SPA, addition of SPA be helpful?
Yeah, excellent question. It's something we're very excited about asking. I can say, having worked in CAR T cell field for a while, I think the SPA is quite distinct in terms of the potency benefit that it provides. We've benchmarked it to a number of other canonical molecules in that sort of class, if you will, and we think it's really distinct and really compelling. So we are excited about potentially applying that in other indications. Maybe one quick point, it is true, in the data that I showed, if you take away the SPA, the cells look quite poor. I think that's actually a bit of an artifact of the system. And so, as you know, in these xenograft models, as you titrate cells up or down, eventually you fall off a cliff, essentially, and you have no activity at all, right? And sometimes that is a pretty steep drop from having some meaningful anti-tumor activity to nothing at all. And we've shown in other models when we don't have the SPA on board that the individual components, the other components add benefit, but the SPA is so potent that you have to be way out on your dose de-escalation, essentially, to even really have a well-controlled experiment there. And so it can make it appear as if the remaining components are worse than they are, I would estimate. But it's very difficult to do the large dose finding study, to put all of those doses in the exact same study to demonstrate the individual contributions.
you have a sense of how PSMA changes after TKI and whether it's still highly expressed?
Yeah, it's a fantastic question. We have not fully answered it, and we are interested in working with some of you in the audience eventually to try to answer that question.
And then is there any functional imaging built in, like PSMA PET or CA9 functional imaging?
Also an excellent question. It's something that we're exploring. It's early days, but it is something that we're exploring.
Just when in prostate cancer, when we look at PSMA imaging, you know, it's much, much higher in salivary glands and liver and whatnot than what we see even in prostate cancer, much less the vasculature and renal cells. So will you get a lot of sort of non-specific binding in these other tissues and maybe not get enough concentrated in the tumor? Are you worried about that?
Specifically for the PET or for the therapeutic T cells themselves?
For the actual, yeah. So like,
Yeah.
when this is preferentially binding PSMA in these non-target tissues.
It's a good question. I think we'll have to see the experiment. I would say our level of concern is not too high, but we'll have to just generate the data.
Anyone else? Otherwise, I would say we move on to the next two speakers. So thank you so much for coming and presenting. Wonderful presentations. Fantastic. Fantastic, yeah. Fantastic. Pavlos is here.
I think I know this guy. Next up is Pavlos Msaouel, who's going to be discussing some efforts with Boston Gene. Is that right, Pavlos?
Indeed, yes. Okay. Thank you for the opportunity to talk about our collaborative efforts through the MD Anderson Boston Gene Alliance. These are my disclosure. I serve as an investigator in collaboration with Boston Gene, but I don't receive any funding or have any COIs with them. And as mentioned yesterday, again, we are very passionate and focused about renal medullary carcinoma, RMC, which is a deadly kidney cancer defined by the loss of the SMARCB1 tumor suppressor. As I mentioned again yesterday, as a reminder, 1995, when it was first described, median survival 4 months. This has improved to 13 months. We set up the benchmark goals to get to 2 years by 2025. Just submitted the proofs of this figure showing that we're steadily heading towards achieving that goal. And now we have to get to 5 years, the median survival by 2030. I mentioned one key aspect yesterday, the MARC16 trial. Today, I'm going to talk a little bit more about other parallel efforts that will get us there. A key way to do this is and has been the 4 dedicated investigator-initiated trials that we did for renal medullary carcinoma and that we have activated since 2018. 3 of them have been completed. 1 is opening, as mentioned yesterday. And it is the biological insights that guide this trial development. That is key. And one of the things that we learned the very hard way, and we published in 2020 in Cancer Cell, is that the standard NGS assays are really inadequate for RMC. You can see in red there how low SNV mutation burden RMC has. And in contrast, what it does have is a lot of focal and arm level copy number alterations. What this means is that even though the SMARCB1 tumor suppressor is lost in 100% of cases, because also of the low tumor purity, the standard NGS assays in less than 10% of cases will identify any mutations in SMARCB1. So you have no idea how many times I hear, from community oncologists, oh, the NGS did not find a SMARCB1 mutation, so it's not RMC. No, it is RMC. Do the IHC first, and then, we'll talk. And nowadays, with the latest versions of the standard NGS assays, the number is about 15 to 20% that will detect a SMARCB1 mutation. And so we need to do better. And so what got us excited about working with Boston Gene is that they have this multi-omics approach that allows this multi-omic discovery, CLIA certified. They do whole exome sequencing that has very good depth along with transcriptome analysis, whole transcriptome, and that is key. That is what we had found in my lab is very, very important with delineating the biology of RMC. It allows as well, you know, to do a very useful TME analysis as well as other goodies that can be used like liquid biopsy, etc. So it's always the key. The key thing is trust but verify, okay? So what we wanted to see is, can this Boston gene assay detect, first of all, the mechanisms of SMARCB1 loss using the exome sequencing? And you can see here that we get some good numbers in the one allele and good numbers even in the second allele. It's very, very similar to what we found in 2020 in our publication in Cancer Cell, but we used a lot of different orthogonal assays to find this. You can see here that the Boston gene did not detect the translocation. Those are very, very difficult to find with WS. We had to do FISH to find the inactivated translocation of the second allele, but they found the deletions, and that is key. So that made us more confident about its ability to help us with biological discovery. I mentioned yesterday the MUC16 expression identified in my lab originally, and again, we wanted to see at the transcriptomic level now what can we find with Boston gene. You can see here the significant upregulation, 60% of cases in the Boston gene cases that we analyzed, 15 patients with RMC that we worked together, consistent with our internal MD Anderson data, and similar to ovarian cancer, we can compare using the Boston gene pan-solid now assay. Now that we know that at least, you know, we have some validation, we can trust this data, now let's look at other pathways of interest. Again, trust but verify. So we had published in Cancer Cell, and this was also verified by our colleagues at MSK-CC, that there is this heterogeneous CD70 expression in RMC as much or close to clear cell renal cell carcinoma. We found this in the Boston gene assay, related, you know, to the ENPP3 targeting just mentioned a few minutes ago. Wanted to see, is this a target for RMC? Doesn't appear to be. Useful knowledge. So this is not, you know, a target for RMC. What we did find is some other pathways of interest, like the HIPPO-YAP. We had gotten a DoD KCRP award with Gianni Genovese to investigate that pathway. It was very interesting to see it being validated here. But we had this suspicion with Gianni that this HIPPO-YAP activation in RMC is due to NF2 loss, but I mentioned it's not an easy thing genomically to detect NF2 loss with standard assays. It takes a lot of work, but with Boston gene, perhaps we can do it. And why did we believe that RMC has NF2 loss? Because NF2 is right beside SMARCB1 on 22q. And what did I say? Large deletions are what we find in SMARCB1. NF2 may be lost as well, and the NGS assays will not be detecting it. And lo and behold, we started finding with Boston gene NF2 loss, copy number alterations that may explain why we're seeing this HIPPO-YAP upregulation, creating another potential therapeutic target for RMC. Now, I mentioned the exome data, let's talk now about the transcriptome stuff. Okay, so you have this potentially valid transcriptome analysis. ADCs make sense for medullary carcinoma. A cancer that can respond to cytotoxics makes much more sense than potentially other subtypes. Let's look into it. One of the key things that we showed in 2020 is that RMC is different than urothelial carcinoma. And so this helped a lot move the field forward, and you can see it here. You can find HER2 being expressed in some urothelial carcinoma cases, essentially none of our RMC cases. Trust by verify in my lab, we looked the HER2 IHC in more than 15 RMC cases, 0 clear certified, consistent with the Boston gene data. Nectin-4, pathway of interest. There is even a trial that is enrolling RMC patients. Let's look it up. You see here, not really expressed in RMC, but here is another interesting target, CD86. Makes sense. Cadherin-6 is associated with kidney morphogenesis. It is expressed in clear cell kidney cancer and other kidney cancers, and it is expressed in RMC. Oh, now we have a target of interest. And now another one, TROP2. Interesting stuff here. You can see a bimodal distribution in the RMC cases. You can see it here compared with the Boston gene pan-cancer assay, this bimodal distribution. Trust by verify. We did the CLIA-certified TROP2 IHC at MD Anderson, and we're seeing, oh, there is this in distribution as well at the protein level. And now let's do the bench-to-bedside translation that, we like to do in this case. 23-year-old, this is an example of a patient that, we analyzed with this 23-year-old patient, classic RMC, metastatic in multiple areas, got the standard therapies we're giving at MD Anderson, carbotaxol first-line therapy, 4 months, typically aggressive RMC, gemcitabine, doxorubicin gives 2 to 3 extra months on average. The panitumumab regimen that we introduced at the KCRS24, and we've now treated more than 25 patients, gives a good punch, 11 extra months. Now we're getting, to the 18 months, 2-year survival. Then we give bortezomib plus tazemetostat 2 months, high TROP2 expression of the Boston genome analysis, started repurposed sacituzumab, and we got a partial response in this patient that happened a little bit more than 3 months ago. Patient is doing very well, and we've treated more patients than that since then. And so in conclusion, Boston Gene is a multi-omics platform that provides reliable biological insights with immediate translational implications. It helps us identify and validate therapeutic RMC targets. I'm giving you a portfolio. People can now jump in, help with our research, do trials, collaborate, or do independently. Here are targets. Let's push it. Let's get the median survival to 5 years by 2030. And there is ongoing work, as you can imagine, to replicate this RMC bench-to-bedside paradigm across smart myelopathies with our first goal, epithelioid sarcoma. You can already tell by yesterday's trial. And our other one is smart beyond deficient sinonasal carcinoma, a very aggressive, deadly head and neck cancer described for the first time less than 10 years ago. Very interested. We found the physician champions, and we're doing research in collaboration with Boston Gene to attack this one as well. And of course, for the audience here, rare and common RCC subtypes. Let's go, let's do work, let's open trials dedicated for the rare RCC subtypes. It's feasible, we can do it even when they're negative. We're getting knowledge from this, and it takes a village to do this. I would like to acknowledge the many people within MD Anderson and my lab in particular. During the hurricane on Monday, some of them were in the lab. They're that dedicated to work. They're totally crazy like me, I guess. And the Boston Gene team that has been our great collaborators through this effort, and of course our funding agencies, and happy to answer any questions you guys might have.
Outstanding, outstanding. And last but certainly not least, Kim Allman from Telix. Thank you so much.
Thank you, Hans, and thank you to the KCRS for the opportunity to present today. So just for a brief overview, Telix is a global leader in radiopharmaceuticals who is developing theranostics for the treatment of oncology and rare diseases. Our theranostic approach is isotope agnostic using clinically validated targets. You can see at the bottom of the slide, the company's motto is See It, Treat It, which is achieved through the development of tumor-specific tumor-specific small molecules and monoclonal antibodies, which are radio-labeled with a diagnostic radio tracer or swapped out for a therapeutic payload. Off to the right-hand side, you can see everything that's going on in development, and we will be discussing what's going on within the kidney cancer space specifically today, but you can see that there's development in multiple other disease spaces, including other GU malignancies. So many in the audience are likely very familiar with the results of the phase 3 Zircon study, which was presented by Dr. Shuck at ASCO GU last year. But for the uninitiated, it evaluated a CA9 targeted monoclonal antibody, girentuximab, which is radio-labeled with zirconium-89, for the non-invasive investigation and evaluation and characterization of clear cell RCC. So you can see the results here of the primary analysis for the ITT. There was a mean sensitivity of 85.5 and a mean specificity of 87% with a positive predictive value of 93%. And these results were actually enhanced in the clinical T1A patient population. This radiotracer, immune PET radiotracer, does have an acceptable safety profile consistent with other radiolabeled antibodies and will be evaluated in the metastatic setting for clear cell RCC in an upcoming phase 3 trial called ZERMET. So here you can see graphically depicted some images from zirconium girentuximab trials. Off to the far right-hand side, we can see an image from the Starburst trial, which is a trial of CA9 expressing tumors outside of clear cell RCC. As we know, CA9 in the hypoxia signaling pathway to play an important role in multiple different tumor types. Within the center, we can see images from the Starstruck trial, which is investigating not only CA9 expressing tumors, but a dedicated clear cell cohort. Off to the left-hand side, we can see images from the Zircon study, where you can see very clear uptake into the right renal mass and also a right posterior rib lesion. So by removing the zirconium-89 radio label and attaching a lutetium-177 payload, this allows for specific tumor targeting with internal radiation while limiting radiation therapy exposure. And by irradiating the tumor cells, this could potentially increase the checkpoint inhibitor response with their immunogenicity. By the direct cell damage and death of these CA9 expressing clear cell cells, this can lead to additional tumor-specific antigen presentation and can facilitate recruitment and infiltration of immune cells into the tumor microenvironment. By further stimulation of the cytotoxic T cells through PD-1 inhibition in the setting of this increased antigen presentation and immune cell influx, this suggests the potential synergy between checkpoint inhibitors and lutetium-labeled girentuximab. So I'm just going to review 3 studies that are in development with you. So Starlight 1 is a frontline trial of lutetium girentuximab in combination with cabozantinib and nivolumab. This is a single-arm phase 2 study, which is evaluating clear cell patients, as I mentioned, in the frontline setting, and enrolling up to 100 patients. This is going to be opening at the end of this month at MD Anderson and will be led by Dr. Eric Yonash, with a couple of additional sites coming on board shortly thereafter. These patients will receive lutetium girentuximab for up to 3 cycles based on disease response and tolerability. These cycles are spaced 12 weeks apart, and each of the subsequent doses are administered at 75% of the preceding dose. Cabo-Nivo will begin on week 5 of this trial, and the primary objective of the trial is safety and complete response. Starlight 2 is an ongoing trial of lutetium girentuximab in combination with nivolumab. This is a single-arm phase 2 study, which is open and enrolling at Memorial Sloan Kettering and is led by Dr. Feldman. This is also an advanced clear cell population who has received at least 1 prior line of therapy, with 1 line having to have been a PD-1 or PD-L1 inhibitor. Similarly, these patients will have the opportunity to receive up to 3 doses of lutetium girentuximab, and that'll be administered in combination with nivolumab, which will start on cycle 1, day 15. There is a safety lead-in and dose escalation for the initial portion, which will enroll 9 to 12 patients. And after an MTD is established, it will move on to phase 2 dose expansion, where if at least 1 patient has experienced a response, an additional 19 patients will be enrolled. The primary objective of this study is safety and MTD, as well as best overall response at 24 weeks. And then lastly is the Starstruck trial. This is a combination of lutetium girentuximab with an oral DNA PK inhibitor. That's a Merck KGaA asset called peposertib. This is a phase 1b dose expansion study that is enrolling patients with CA9 expressing tumors. These patients also will have the opportunity to receive 3 doses of lutetium girentuximab with an optional 4th dose if they do have a response per RECIST criteria. This will be given in combination with peposertib, which is dosed on days 4 through 21 of an 84-day cycle. In the dose escalation portion, there'll be 15 to 20 patients enrolled, and then moving on to a recommended phase 2 dosing where there will be a cohort B who will enroll 20 patients that are CA9 expressing, and then an additional cohort A, which will be clear cell RCC patients only. The primary objectives of this study are safety, tolerability, recommended phase 2 dosing, and looking at several secondary endpoints of efficacy. Thank you for your attention.
Good, we're open for questions. I have a question. Zai, if you take the microphone. I have a question for Pavlos.
Sure. Okay.
Okay, in a second.
Yep. Oh, sorry. Question for Kim. Kim, how was the dose picked, selected for monotherapy first, and what was the safety of, your drug as a monotherapy?
Sure.
Do you have some data?
Yes. So there was a phase 1 study, which was a safety study and dose exploration. So the patient started at 1,100 megabecquerels per meter squared, and it increased in doses of 370 megabecquerels per meter squared in a dose escalation format. So it was an ascending dose study. So what was found is 56% of patients received 2 doses, and then 17% of patients were able to receive 3 doses. So after the patients had received their first dose, about 70% of patients had stable disease, with one patient having a partial response. And the disease trajectory coming in for this phase 1 study, the growth of the tumors on average was a mean of 40.5%, and then ended up plateauing out to 5% growth after the first administration. So you mentioned about the dose-limiting toxicity, and that was myelotoxicity, which ultimately led to the dose that was selected as the monotherapy. But the doses that are in both of the Starlight trials as well as the Starstruck trial are lower, because this is a dose-limiting toxicity, particularly from a thrombocytopenia standpoint.
So just follow-up question. There were some manufacturing issues with this. Have you resolved the manufacturing issues?
Yes, and those are ongoing efforts too, as it's kind of a complicated process to radio-label the antibody and get it out. But it does have a long half-life, which allows for a lot of stability.
You do have a microphone?
Yes. Great presentations. Both the presentations are awesome. So Pavlos, has MUC expression been checked in tissues?
MUC16?
Yeah.
Absolutely. And there is a question by in the chat as well. So I showed a slide yesterday showing that there is, because a question is, has CA125 been observed in the serum? Yes, we've checked in more than 40 patients with RMC formally at MD Anderson, and we have found that it correlates very nicely with MUC16 levels by IHC. So when you get IHC 3+, you can get CA125 up to 2,000 units per ml, like huge levels, 3,000, 4,000, etc. That's why our MUC16 trial that is targeting cell surface MUC16 is going to use serum CA125 as its initial criterion, and only if they're negative in the serum will we check. I mean, we will check in general, but we will use as eligibility criterion the MUC16 levels in the cell surface. And there is some interesting heterogeneity connected with morphology. From a pathology perspective, we can talk more about that afterwards.
Sorry, 2 more quick questions. So is the converse true? Like we are seeing, like you have mentioned, NF2 events in SMARCB1 deficient cases, but now we also have NF2 deficient renal cell carcinomas. And you think, is there a chance that SMARCB1 deficiency might be a secondary event in those? And the last was, so there is an advent of CRISPR therapy now in sickle cell disease. And do you believe or do we know, or do you believe that that can reduce the chances of metastases or disease aggravation? Because some of the carcinogenesis is happening because of these vascular events. Thank you.
Boy, what a brilliant question. So I will tell you that we have analyzed a few formal NF2 mutant cases, like the ones you're thinking of, and one of the cool things that we're looking forward to doing is compare and contrast them with the SMARCB1 deficient NF2 mutant. There is a case report where we have a patient that has what we call RCC-UMP. This is the rare subtype of RMC that does not happen in sickle hemoglobinopathies. That's 6% of the total of RMC cases. In our case series that's going to be published soon of 135 patients with RMC, only 9 were like that. And one was that case report that we published that has neurofibromatosis type 2, NF2 germline, along with meningiomas and other tumors, and developed a SMARCB1 deficient RMC, suggesting again the connections. That same paper that is going to publish also finally answers the question with data: what are the electrophoresis patterns of patients with RMC? Are there differences in hemoglobin F and other levels that could help guide potential, preventive strategies, etc.? It appears that, and you'll see that in the discussion of the paper, that patients with RMC have average to high levels compared with the average population with sickle cell trait. So they don't have low levels of HbF. So they have the protective effect of HbF. Now it's our job to look mechanistically whether they need more of that or we need to target other pathways in the hopes of prevention.
Yeah, first, I really love this session. Thank you very much. So my question is for Kim. You showed the data of your popersertib combination trial. Did you have any preclinical data with this agent or other radiosensitizing agents to share?
Yes, I don't have it readily available, and I'm newer with the company, so I can certainly share it with you at a later time. But there has been preclinical work that's been done, and this study is being exclusively run in Australia right now.
Yeah, Pavlos, so there's the INI1 deficient tumors that have high level of PD-L1 expression. Is that a different type of tumor than the typical medullary tumors that are occurring in the sickle cell patients?
Beautiful question. So one of the first times that we realized how nicely the transcriptomic data, surprisingly, in RMC correlate with the protein level was in our 2020 cancer cell paper. We show at the RNA level that there is a lot of heterogeneity in PD-L1 and PD-1 expression with, I would say, at least 1/3 or more of formal RMC cases having very high levels of PD-L1 and PD-1 both at the RNA level, bulk RNA-seq at the time deconvoluted, protein IHC done with our immunotherapy platform with Pam Sharma, and now we have single-cell sequencing data showing that. We know, and we knew since that paper, that RMC is naturally a hot tumor. That's the difference, let's say, with chromophobe that is a much, much more cold tumor. It's in theory as hot as clear cell RCC, but really, really, really does not respond to the standard immune checkpoint therapies. And we're working to find out why. And that's part of the rationale for yesterday's trial, because at least one resistance mechanism that we are seeing orthogonally is the downregulation of MHC class 1 that we hope will be bypassed by the CD3 by MUC16 bispecific.
Yeah, and the second question, I think one of the kidney cancer cell lines that gets used a lot, SN12, is NF2 mutant. Is that an RMC line?
No. So that's, and it's connected, or at least I don't think so. It's connected to what Rohit also asked. What is the role of NF2 loss? It seems like we're seeing NF2 loss in RCC a little bit more than we expected. You know, we have the formal NF2, the biphasic hyalinizing carcinoma that Rohit has done a great job characterizing. So it has a very specific morphology. You can see it. But then there is this unclassified more RCC, although Rohit may be observing certain parts with better classified. There's also NF2 mutant, formally NF2 mutant by SNVs. But at the same time, now we have at least RMC that also appears not to have SNVs, but copy number losses that appear to upregulate TPO-YAP. And I can tell you that we may also be seeing in some of the other distal nephron cancers like HLRCC. Something about the biology potentially of the more distal nephron tumors, perhaps we can brainstorm this, seems to like that pathway more than expected. And that is a great opportunity for therapeutic targeting, because a lot of people mistakenly, including a lot of companies, mistakenly think that, oh, I am going to target mesothelioma. But we may have really great therapeutic opportunities in the kidney cancer space across subtypes.
Thank you. This sort of lends itself to the conversation we were having this morning around early drug development. But you know, you take a patient as you describe in your scenario, and you're so helpful in helping us out in the community, in terms of treating these folks. Patient's gotten platinum up front, anthracycline chemotherapy, right? Then after that, perhaps EGFR-directed therapy as you've prescribed. And at that point, I mean, do you imagine elements like MUC16 are stable targets? I mean, think about all the modifications that mucins go through, right, with therapy, glycosylation, et cetera.
Beautiful question, and we're looking into it even more deeply, because remember I mentioned yesterday exactly that SMARCB1 loss is necessary but not sufficient for MUC16 expression. There's a lot of interesting biology happening there. Thankfully, as mentioned before, CA125 levels correlate well with cell surface MUC16, and I can tell you that it appears that throughout the treatment course of these patients, they continue to express serum CA125. So a patient that goes to the first line, second line, third line, it correlates very well with tumor burden. As soon as they become resistant, you see elevated serum CA125 levels that are potentially therapeutically targetable.
Beautiful. Any last question? Otherwise, I will turn over to Brian. So thank you so much, everyone.
Thanks.